Before a nerve pulse occurs, the axon is at resting potential, which is negative 65 millivolts. Once the axon gets stimulated, the sodium gates open and sodium floods down as concentration gradient into the axon. As sodium floods in, the membrane changes from negative 65 millivolts to positive 40 millivolts, and the impulse fires as it has passed through the threshold of negative 40 millivolts. This process is called depolarization because the charge of the axon changes from negative to positive. Secondly, the gates of potassium gates open and potassium moves outside the axon, changing the polarity from positive 40 millivolts back to negative 65 millivolts. This is called repolarization because the inside of the axon resumes a negative charge as potassium leaves the axon. As soon as an action potential has moved on, the axon undergoes a refractory period, during which the sodium gates are unable to open. Therefore, the action potential cannot move backwards and instead always moves forward down an axon toward its terminals. During refractory period, the sodium-potassium pump has to restore the previous ion distribution by pumping 3 Na to the outside and 2 potassium to the axon, and the ions in the axons are restored to its previous state. As soon as the refractory period concludes, the axon is back to its previous state and the cycle can continuously repeat over and over.
The nerve impulse begins at the dendrite of the cell body and then moves along the axon to the synaptic cleft, where then it jumps to the next dendrite of the cell body to repeat the process once again.
Every axon branches into many fine endings, each tipped by a small swelling called an axon terminal. Each terminal lies very closely to either the dendrite or the cell body of another neuron. The region of close proximity is called the chemical synapse. It is important to note that two neurons at chemical synapse don't ever physically touch each other. Instead, they are separated by a tiny gap called the synaptic cleft. At a synapse, the membrane of the first neuron is called the presynaptic membrane, and the membrane of the next neuron is called the postsynaptic membrane. Communication between the two neurons at a chemical synapse is carried out by molecules called neurotransmitters, which are stored in synaptic vesicles in the axon terminals. When nerve impulses travel along an axon, reach an axon terminal, gated channels for calcium ions open and calcium enters a terminal. The sudden rise in calcium stimulates synaptic vesicles to merge with the presynaptic membrane, and neurotransmitter molecules are released into the synaptic cleft. They diffuse across the cleft to the postsynaptic membrane, where they bind with specific receptor proteins. Drugs can affect the synaptic transmission in different ways. One example is cocaine, which invaginates the neurotransmitters and engulfs them, preventing the receptor proteins from receiving neurotransmitters.